secrets. Everything gets like, I was like, oh, my undergrads. I just have one black one. The undergrad. Does anyone else have the black one?
Okay, should we resume our discussion? Are there any questions that came to your mind over the break?
Free stuff. Yeah, I like free stuff. It's like soothing. It's like sleeping by ocean.
Oh, no, you're good.
Okay, questions? There was one question during the break about when urea cycle activity was highest. Generally, if you have a typical high carbohydrate meal, it's going to be very low in the fed state, but we know that fasted metabolism after an overnight fast, that's where you're doing all that amino acid metabolism, right? So that's when urea cycle activity is highest. If you prolong the starvation for longer than that, your body is going to switch to fatty acid metabolism, and the urea cycle activity is going to go down again. Okay. Okay. Let's see. Okay, so just sources of nitrogen for the urea cycle. The free ammonium, this comes from deamination. For example, the glutamate dehydrogenase or the glutaminase reactions, or from AMP deamination. And then the aspartate, this is taking nitrogen from transamination reactions. Nitrogen that's going through that alpha ketoglutarate glutamate conveyor belt. Okay. Okay, got your turning point thing ready?
Not open yet.
What? Sorry. Good.
Oh, nice.
Okay, this should be 100%. You think? Hey. All right, I'm done. Okay, so within the urea cycle, disorders in the enzymes in the urea cycle, also in that ornithine citrulline antiporter, they all manifest as hyperammonemia. And so you knock out the urea cycle, you're going to have increased amounts of ammonium, and it's going to cause toxic effects. And that's going to manifest as primarily neurological symptoms. There are some reports where a mild presentation of a urea cycle disorder can manifest as a protein aversion, or just, they know when they eat a lot of protein, it makes them feel bad, so they try to avoid it just kind of naturally. Conversely, urea cycle disorders can be exacerbated by eating high protein meals. So if you increase the amount of protein in the body, that's going to exacerbate the problems with the urea cycle disorder. Okay, so clinically, how do we know when somebody has hyperammonemia or not? And you might consider one of these enzymes can use free ammonium as a substrate. Okay, very good. So glutamate dehydrogenase is used in the clinical assay to measure ammonium in a patient's blood. So we take a patient's blood sample and we want to know how much ammonium is in it. So the patient sample supplies this, and then the company that makes the assay, they supply a recombinant glutamate dehydrogenase enzyme, a bunch of reduced NADPH, and alpha ketoglutarate. So you take a patient's blood sample, you add the alpha ketoglutarate, the NADPH, and the recombinant glutamate dehydrogenase enzyme. The reaction goes this way to make glutamate, and the more ammonium is around in the patient from the patient's blood, the more the reaction is going to go this way. What's actually being measured is the NADPH concentration. NADPH has a specific absorbance that's pretty easy to measure in a spectrophotometer. So the assay is measuring the rate of decrease of NADPH directly. So this would be the case if you wanted to measure free ammonium in a patient's blood. If you wanted to measure blood urea nitrogen, or how much of the nitrogen was in that urea form, you would have a patient's blood sample, and first you would incubate it with urease. And this is a bacterial enzyme. We don't make this, but some bacteria have a urease enzyme that can cleave the nitrogen from urea. So you first do this reaction, and then you couple it with this reaction, and there you get the blood urea nitrogen. Okay, let's see. So let's segue to urea cycle regulation. Gesundheit. Okay, good. Arginine is acting as one of the major regulators of the urea cycle activity through that N-acetyl glutamate synthase. So carbon-whale phosphate synthetase, that's the first enzyme in the urea cycle that's making that bond between bicarbonates and ammonium and phosphate from ATP. Carbon-whale phosphate synthetase 1, this is allosterically activated by N-acetyl glutamate. N-acetyl glutamate is made by N-acetyl glutamate synthase, or NAG synthase. So that takes the amino acid glutamate, and it takes the 2-carbon acetate from acetyl-CoA, and it adds it onto that amine group to make N-acetyl glutamate. So arginine is acting as an allosteric activator of N-acetyl glutamate synthetase. N-acetyl glutamate then acts as an allosteric activator of CPS1. So arginine is indirectly serving as an activator of CPS1 activity, and that helps to regulate or increase the amount of substrate flowing through the TCA cycle, the urea cycle. Sorry, forgot what day it was. Okay, so arginine is, if we have a lot of arginine around, that's both going to increase the activity of arginase just from because it's a substrate, Gesundheit, and arginine is also going to activate CPS1 indirectly through that allosteric activation of N-acetyl glutamate synthase. So I think in your objectives it talks about 3 ways that the urea cycle is regulated. So arginine activating arginase is one, arginine activating NAG synthase is another, and then glutamine activating transcription of urea cycle activities is a third one that I'll give you a slide for that in the quiz slides downloads, but it's again, it's a little esoteric, and it occurs when you have a huge amount of protein and excess nitrogen around. You get a long-term transcriptional remodeling of the urea cycle enzymes. Okay, so let's move on to urea cycle disorders. If you start with the CPS1, there are 5 enzymes in the urea cycle. There's also a 6th here, NAG synthetase. And I should have put a number 7 here for the ornithine citrulline antiporter. So inherited mutations in all 7 of those proteins can manifest as urea cycle dysfunction and hyperammonemia. Okay. I'm not going to go through each one of them because basically their clinical presentation is pretty similar, but how you diagnose them and the treatments are going to be somewhat different. So I'm going to talk about how we do how you would diagnose which enzyme is defective in the case of hyperammonemia for a few different cases, and hopefully, and then we'll run through a couple of clinical cases at the end of lecture, and hopefully it'll be pretty clear. Basically, if you suspect a urea cycle disorder, you're going to look for amino acids that are elevated in the blood. And if one amino acid is elevated, for example, if arginosuccinate is highly elevated, then the enzyme that's immediately upstream of it, that's the likely problematic enzyme. Okay. So that's one strategy, and I'll elaborate on that in a bit. Another strategy is to look for orotic acid in the urine. Now, there are 2 carbamoyl phosphate synthase enzymes. The CPS1 is mitochondrial and is using inorganic nitrogen in the form of ammonium as a substrate. The CPS2 is important for pyrimidine synthesis. That's a cytoplasmic enzyme, and that's using glutamine as a nitrogen donor. Okay, so they can intersect in the case of a urea cycle disorder, and the urea cycle disorders that are proximal to the generation of carbamoyl phosphate. So just as an example, if we have a deficiency in the ornithine transcarbamylase enzyme, carbamoyl phosphate will build up. That will then leave the mitochondria and intersect with that pathway in pyrimidine synthesis that ultimately is going to make uridine monophosphate and other nucleotides. The intermediate orotic acid, or orotic acid, excuse me, that will, okay, so if you have excess carbamoyl phosphate from the mitochondria, that's going to divert out into this pathway. However, there's feedback inhibition of the nucleotide synthesis pathway, right? So you're not going to get all of this bottom product. You're going to get the intermediate orotic acid that will build up. This can be detected in the urine, and the higher the orotic acid is, the closer to carbamoyl phosphate the defective enzyme is. Does that make sense? So if you have an OTC deficiency, that's going to be have a very high level of orotic acid in the urine. If you had an arginine lyase deficiency, then these intermediates, they have a capacity to go out and divert into other metabolic pathways. So you're not going to get as much carbamoyl phosphate built up. And in the case of a more downstream urea cycle disorder, you could have a small elevation or no elevation in the urinary orotic acid. Okay, is that clear? Okay. Okay, so one way we can diagnose urea cycle disorders is to look for urinary orotic acid. And another way we can do it is by looking at amino acid elevations. So you can measure so you'll measure hyperammonemia first. Hyperglutaminemia is frequently also observed in urea cycle disorders, so glutamine will be elevated as well as the ammonium. But if you're looking at other intermediates like arginosuccinate or citrulline or ornithine, you can measure those concentrations. And as I mentioned, if you have a huge amount of citrulline around, then that suggests that the deficient enzyme is arginosuccinate synthetase. Okay, so if you measure all of these amino acids and citrulline is the one that is the most out of normal, then it's likely that this is the defective enzyme. And you may or may not also see a decrease in the product of that deficient enzyme. Yes?
What if there's a problem with the transporter? Like, what would that look like?
That results in HHH syndrome. So you get, and that's a great segue, so that's the next slide. So deficiencies in the transporter result in HHH syndrome, and the HHH syndrome stands for hyperammonemia. So you get elevated nitrogen or elevated ammonium, and that's not unique to this condition, but that's a universal feature of urea cycle disorders. You also get I think I've got each one of these, so I better. The ornithine, this is trapped in the cytosol, and the hyperornithemia results because the ornithine can't get out of the mitochondria and it become a substrate for the ornithine transcarbamylase. The homocitrulline, so hyperornithemia, hyperammonemia, and then homocitrullinemia. The homocitrulline, this is generated when the carbamoyl phosphate doesn't have the ornithine around to be a substrate for the ornithine transcarbamylase. In this case, the carbamoyl phosphate bonds to lysine and becomes this homocitrulline. And this homocitrulline is, I don't believe it's toxic, but it is something that is not around in great amounts except in the context of this HHH syndrome. So the basic presentation is going to be the same. Initially, you get the high ammonia and the consequences thereof, the neurological symptoms that result from it. But the specific pattern of amino acid elevations is going to be distinct for this condition. Okay, so Nelson's Textbook of Pediatrics provides a pretty nice diagnostic key for diagnosing an infant with hyperammonemia. So basically, we kind of go down this route. So obtain plasma amino acids. If there's a specific amino acid elevation, then you can diagnose one of these conditions. If there's not a specific amino acid elevation, then you would get the orotic acid in the urine. And if that's high, that would suggest an OTC deficiency. If it's normal or low, then you look for the citrulline. If that's low, then that can be the NAG synthetase, or either a deficiency in the NAG synthetase, that enzyme that generates the allosteric activator of CPS1, or a deficiency in CPS1 itself. If the citrulline is normal or it's high, then this is just hand waving, we don't know what's wrong. Hopefully it goes away. And I've got a couple of cases that I think we can go through. I'm going to talk about treatment of urea cycle disorders for a bit, and then we'll go through a couple of clinical cases and kind of show how this diagnostic key can work. Okay, so the goal of the treatment with urea cycle disorders is to get rid of the excess nitrogen. And that may be, acutely you might have to rehydrate or take other measures to get rid of the nitrogen acutely. But once that's resolved, you need to get rid of the excess nitrogen that's going to be generated on a daily basis. So low protein diet is the kind of the easiest thing that you can do for the urea cycle disorders, and just try to eliminate the potential for ever being in positive nitrogen balance. N-carbamoyl glutamic acid, this acts as acts like N-acetyl glutamate, and this can activate the CPS1 that can compensate for a deficiency in NAG synthetase. It can also potentially compensate for mutations that result in partial loss of function of the CPS1 enzyme itself. You can also eliminate nitrogen in alternative pathways. So arginine, the amino acid arginine, is used in a couple of specific conditions, arginosuccinate lyase deficiency and the HHH syndrome. Benzoic acid and phenylbutyrate, these are molecules that will condense with other amino acids and make urinatable products. So it's basically bypassing the urea cycle and allowing nitrogen to be excreted through alternative products. Liver transplant, this is something if you recognize it too late, or if the liver is totally crapped out, then you have to take more extreme measures. Viral transduced gene therapy, this is still more aspirational than actual. However, there was a kid who was, maybe 20 years ago or so, who was given a fulminant leukemia as a result of attempting to correct OTC disorder with viral transduced gene therapy. And it was a pretty famous case, and it kind of threw a lot of cold water on the idea of using viruses to correct inherited disorders of metabolism. However, I think there's still potential here, but there's some practical reasons why it's there are problems with it. You could give a patient something else besides a urea cycle disorder that could be potentially much worse. Okay, I'm going to discuss the benzoic acid and phenylbutyrates first, and then I'll touch on some of those other treatments. Okay, so just conceptually, the normal urea cycle functions thusly. We take proteins in our mouth, and each amino acid has a single dot representing a nitrogen. Those amino acids go through the to become either aspartate or free ammonium substrates for the urea cycle. So the urea now has two dots, each dot is a nitrogen, and the urea goes out through the urine. Okay, if this doesn't work, or if we have a deficiency in the ability to make urea from the amino acid pool, phenylbutyrate and benzoic acid, these can be used to eliminate nitrogen as in alternative ways. Phenylbutyrate, this drug, it's a phenol group with a short chain fatty acid, butyric acid, essentially on it. Phenylbutyrate gets beta oxidized down to phenylacetate. Phenylacetate, if you'll recall, that was one of the toxic products that builds up in patients with PKU, okay, as an alternative route of phenylalanine catabolism. However, the phenylacetates, in this context, it's making a complex with glutamine to make phenylacetylglutamine, and this is a urinatable compound. Okay, so you can excrete the phenylacetylglutamine out the urine, and each glutamine molecule has two dots or two nitrogen atoms that you're getting rid of. Similar situation with the benzoic acid. Benzoic acid makes a complex with glycine to make hippuric acid. The hippuric acid then is again excretable in the urine. In this case, because glycine only has a single nitrogen, you're not getting as much nitrogen per molecule of the drug out, but still can be functional. Okay, so this is recapitulating a lot of what I just said. I got a little ahead of myself. So the benzoic acid, this gets activated through joining to coenzyme A. The coenzyme A benzoic acid complex then can bond with the amino acid glycine. The coenzyme A leaves, and then the hippuric acid is excretable, and that's got one nitrogen with it. Likewise with the phenylbutyrate, the side chain gets oxidized to phenylacetate. The phenylacetate makes a coenzyme A complex. The phenylacetyl coenzyme A then binds to glutamine. The coenzyme A gets off, goes away, and we get the phenylacetyl glutamine, which can be excreted. Yes?
So the reason that we can excrete that compound in like this situation and not in PKU is because there's more nitrogen? Or is it just that this is like a much lower level of that compound that's given?
I think both of those things are correct. So in the case of an urea cycle disorder, remember, in addition to the hyperammonemia, you're also going to get a hyperglutaminemia or excess glutamine around. So there's going to be more glutamine around in the patients with a urea cycle disorder than with with PKU. Also, I guess I'm not sure of the dosing, and maybe there's not enough of this to become toxic. Kind of my hand waving explanation. Okay, so we've got benzoic acid, phenylbutyrates, arginine. This can be a successful treatment for deficiencies in arginosuccinate lyase. So let's see. So our normal urea cycle is going to go down this way. We pick up a nitrogen from ammonia, then a nitrogen from aspartate to make argininosuccinate. And arginosuccinate lyase cleaves off the urea and regenerates arginine. So if you don't have the arginosuccinate lyase is cutting off fumarate to make arginine, then arginase cuts off the urea to make urea and ornithine. So if you have a deficiency in arginosuccinate lyase, there we go. You can treat that with excess arginine. And the idea here is you're doing a partial urea cycle and blocking or building up of arginosuccinate. So this is a normal intermediate in the urea cycle. This is when it builds up to exceed the renal threshold, that can be excretable in the urine. So if you didn't have this excess arginine, you wouldn't build up enough arginosuccinate to be able to pee it out or to be able to pee out excess stuff. So when you're adding arginine, if you add excess arginine in the case of the arginosuccinate lyase deficiency, you're adding in 2 nitrogens into the body. Those 2 nitrogens go away to make urea, right? So you're not gaining anything really through that part. But as the ornithine goes through the rest of the cycle, it's going to pick up new nitrogens. And if you're peeing out the arginosuccinate, you're getting rid of 2 extra nitrogens in that fashion. Okay, is that clear? It's a little complicated, but okay. So that's arginine, and I'll talk. Oh yes.
So in that case, then you would have really low levels of fumarate because that reaction's not going forward. So you would have to be consuming more arginine, and then you would be excreting more arginosuccinate, and then we'd have lower fumarate, correct?
Yes, except remember that fumarate is not unique to the urea cycle. So there's other sources and destinations of fumarate. So I don't think that you would have a measurable decrease in fumarate. You wouldn't have any produced by this pathway. Yeah, good question. Okay, so we talked about benzoic acid, phenylbutyrate, and arginine. N-carbamoyl glutamate, this can be a treatment for, as shown here, disorders in N-acetyl glutamate synthase. N-carbamoyl glutamate, it's got a similar structure to N-acetyl glutamate, and it acts as a allosteric activator of CPS1. So that can compensate for the loss of the NAG synthase enzyme. N-carbamoyl glutamate, and not shown here, but there are partial loss of function mutations in CPS1. So if you have a mutation that decreases but doesn't eliminate CPS1 activity, the N-carbamoyl glutamate could be a potential treatment for that as well. Okay, so that's all I've got for the treatments. Should we go through a couple of cases? Okay. Okay. 6-day-old male presents with poor feeding and lethargy, and physical examination reveals depressed neurological function. Now, let's say you saw this kid not on the day that you had a lecture on the urea cycle. What would be your next course of action?
Do some labs.
Do some labs? All the labs. Got to check his insurance first, right? That's the first step. Okay, so we have very high blood ammonia and very high blood glutamine. Urinary orotic acid was moderately elevated. So what would be the next thing that we would do? Well, or is this enough to diagnose the patient?
You know what it's not.
What isn't it?
Not OTC. It's not absence of OTC.
Yeah, it's unlikely to be a OTC deficiency because we got moderate elevation of the urinary orotic acid. Okay. So what else would you look for then?
What amino acid is elevated?
Okay, good. So we found arginine is low, ornithine is low, citrulline is high, and aspartate's normal. And carbamoyl phosphate is elevated. Okay, so this is up, this is down, this is up, and this is down. Yeah, so it could be arginosuccinate synthetase or arginosuccinate lyase deficiency. Right? I don't think you could really distinguish between those two based on these data. But if you take skin fibroblasts, so you gotta put a big hole in this kid's arm to get some skin fibroblasts. So they were obtained and grown in culture, and then they were lysed open so that all the enzymes in the skin fibroblasts are in solution. Then they're incubated with aspartate and citrulline and ATP. And if this was a healthy kid, what product should form from that?
Arginosuccinate?
Arginosuccinate, right. But in our kid, no product was observed to form. So based on these data, the patient was diagnosed with an arginosuccinate synthetase deficiency. Now, the urea cycle doesn't really happen in skin, but apparently there's enough enzymatic activity there. I mean, the gene is there. The gene is there in every cell in the body, right? So there's enough of the gene expressed there that you can do this type of an assay to make a diagnosis. So this would be a functional way to diagnose it. You could also diagnose it genetically and look for known or novel mutations in the actual suspect genes as well. Okay, so based on the deficiency in arginosuccinate synthetase, what treatment would you recommend? Low protein diet, yep. What else?
Would you give them arginine? Or like have them arginine or.
No, okay, so would arginine help? Remember, arginosuccinate, this is the intermediate in the urea cycle that is able to be excreted in the urine. So adding extra arginine, you would go from here, here, and you'd be stuck there. So you'd be stuck at citrulline and you wouldn't make the arginosuccinate. So the excess nitrogen, you would have to use benzoic acid or phenylbutyrates to get rid of it. And I don't think that I have the treatment of this case, but the diagnostic flow was essentially this. So they actually did the urinary orotic acid first in this case rather than second as shown by the diagnosis, but really it was looking at the specific amino acid elevations. That would be, and then confirmed the arginosuccinate synthetase deficiency with that assay with the skin fibroblasts. That condition is more commonly called citrullinemia because of the characteristic increase in citrulline in the blood. Yes?
How does growth fit into like the amount of free nitrogen in the blood? Like if a kid's growing really fast, would they be then they need the nitrogen excess anyway? Like how does like if he's 6 days old, he's growing pretty fast, does that mean he would be able to take on more protein than say a full grown adult who's a stable weight?
Yes, so he would be in positive nitrogen balance because he's growing. But he's going to still be going through the fed and fasted metabolic states, right? And in that fasted state, he's still going to be breaking down amino acids or breaking down protein and generating free amino acids, and that has to be gotten rid of. The nitrogen has to be gotten rid of through the urea cycle. So, yeah, I think there might be a higher threshold, I guess, than an adult, but these conditions aren't generally diagnosed as adults. They're diagnosed as in infancy. Yeah, good question though. Was there another question behind?
So in this case, you showed that citrulline was elevated, but CPS1 was also elevated.
The carbomoyl phosphate was elevated.
Yeah, yeah.
Yeah,
typical.
What's that?
Is that like typical for a case because that's the substrate prior to the antiproto step?
This is the only case report that I've seen where they actually are measuring carbomoyl phosphate. So I'm not sure if it's just difficult to measure or why that is or why they did it in this case and not in others.
Relevant for this case?
It's consistent with this case, right? If the citrulline has no metabolic fate, then that's going to product inhibit the OTC enzyme and cause an increase in carbomoyl phosphate.
Okay. Got it.
Yeah, good question though. Okay, should we look at another case? Any impressions? I'll give you a moment to read through it. Okay, let's take a look at some of the laboratory values. So the infant was, 12-month-old? Yep, okay, so diagnosed in infancy yet. So this is before treatment as the kid is hospitalized, and you can see, let's see, the ammonia Definitely not as high as the arginosuccinate synthetase case that we saw earlier, but it's still high, and over time it's getting higher. AST and ALT, this is alanine aminotransferase and aspartate aminotransferase, so we know what those enzymes function is, definitely. And those are used clinically as markers of liver damage. So normally the hepatocyte has a lot of those enzymes in them, and they're supposed to be contained within the hepatocyte. If you have liver damage, those enzymes spill out into the blood, and you measure those activities in the blood, and if they're high, that suggests liver damage. Let's see. So likewise, the gamma-glutarate transferase, that is also similarly is suggestive of liver damage, and more specifically of damage to the name of the cell type escapes me, but it's the epithelial cells that are lining the bile duct as it's going into the liver. International normalized ratio, this has something to do with coagulation time, and it's not prothrombin time, but it's somehow related to that.
So you use prothrombin time to calculate it.
Okay, so so it's derived from the prothrombin time? Okay, thank you. So this is suggestive. If it's high, that means that the cells in the body are not coagulating, right? And remember that coagulation factors are made by the liver. So this is a indication that the kid is at risk of bleeding, and also indicates that there's liver dysfunction. Okay. Okay, let's see. So I got that down there. Okay. So after day 6, further metabolic tests became available, and we found that there was elevated blood concentrations of ornithine, homocitrulline or homocitrullinuria, and urine orotic acid was not measured. Any so what do you suspect based on these data that the child's problem is? This sounds like HHH syndrome, right? Hyperammonemia, homocitrullinemia, and hyperornithemia, right? Any intervention that you would do just immediately, like before you got these data?
Arginine supplementation?
Yeah, you could do arginine supplementation, low protein diet. Yep. Okay, so what did we do? We got the arginine supplementation. The intralipid, so this intralipid and dextrose, this is a like fat and carbohydrate food to give the kid nutrients without any protein around. This Cyclin X1 formula, this is a formula that is devoid of non-essential amino acids. Okay, so any amino acid that the kid can make by herself, are going to be removed, and that's going to kind of remove the excess nitrogen burden on the child. Okay, so in terms of the diagnostic route, we had plasma amino acids, and the specific amino acids led us directly to the HHH syndrome. That one was pretty straightforward. So after that arginine treatment, let's see, the ammonia levels normalized, and so did the liver function enzyme tests. Okay, so arginine, like why you would use arginine for HHH syndrome is a little complicated, and it's not really about the arginine going through the urea cycle as it is with arginosuccinate lyase deficiency. The arginine, let's see, where are we here? Okay, so don't worry about this stuff over here. The arginine can be a substrate for generating ornithine. Ornithine then can generate polyamines, so this would be putrescine and spermine, and these little carbon-carbon nitrogen subunits that are repeated over and over again and they're urinatable. So the polyamines shown here, I think spermine is the main one that is that's generated. The ornithine can also make creatine, and through complexing with glycine, and that then can be excreted as well. So the arginine, that treatment, I wonder if I've got a better view of that here. Yeah, I guess it's a little bit better here. So arginine goes through arginase, which is functional in the child to make ornithine. The ornithine then can make putrescine, and putrescine can dimerize or multimerize to make the spermidine, and spermidine ultimately, or spermine can ultimately be urinated out. So these are the polyamines from that previous slide. The ornithine then can also be a substrate for creatine generation, and creatine is easily urinatable. Okay, so that's a little bit complicated, but just recognize that it's a way to get rid of excess, get rid of extra nitrogen in the body. Okay, should we do a couple of clicker slides? I've got a couple of versions of this question too, so if it's not clear now, it might be in the second version. Okay, slim majority are picking arginosuccinate lyase. So I'm going to ask the same question with the addition of a visual aid. And that makes it a little bit easier, right? So I'm not sure about, why you would measure some amino acids but not others, but I guess there are different panels of these amino acids. So some of them measure everything, some of them just measure a subset of them. Okay, 100%. Close. Okay, should we try another one? A similar structure there. I'll give you the first just the stem and then the stem with the picture.